All right, chapter 5, eukaryotes. Remember, eukaryotes are cells with a nucleus. They've evolved after prokaryotes have evolved into eukaryotes. Okay, okay. So the history of eukaryotes, they appeared about 2 billion years ago. So they've been around for a while. Evidence suggests that they evolved from prokaryotic organisms by symbiosis. In other words, something went inside of the cell, like another prokaryote, and became an organelle in the cell, got trapped inside of it. And they started to develop into eukaryotes and formed a nucleus. So organelles originated from prokaryotic cells that get trapped inside these future eukaryotic cells. And they start to develop organisms, a nucleus, mitochondria, Golgi apparatus, things like that, so they can process different things within them. Okay. This is one of the evolutionary theories of a symbiotic relationship. Okay. So you got a large prokaryotic cell. Okay. You see how the cell membrane kind of vaginates in, forming a nucleus, and it migrates into the center of the cell. And then a prokaryotic cell infects the cell. Oh, I can't write this. Comes into it here, and that gets invaginated into it. And then those start to multiply, and that forms different organelles. And at the end of all this, it keeps developing. Those prokaryotes that infected it now become part of the eukaryotic cell. Okay? And you form different organelles inside, like chloroplasts for plant cells if they're going to do photosynthesis. If a cell wall can be formed, if bacterial cell wall. Okay. Animal cells would form just a cell membrane, okay, with no cell wall. But that's basically the theory of how, through symbiosis, that they formed. Okay. Eukaryotic cells can be unicellular, like a protozoa. They can be unicellular, colonial, or multicellular, like fungi and algae. Okay. They can be multicellular, except reproductive stages like helminths. Helminths are parasitic worms, or arthropods. Those are animal vectors of diseases, like mosquitoes can carry malaria. Okay, so the animals can do different vectors for transmitting that bacteria or the virus to human beings. It's an animal vector for the disease. Okay. This is our eukaryotic cell, typical. They will have lysosomes. Lysosome is an organelle that phagocytizes, or basically eats foreign materials like pathogens, things that enter the cell. The Golgi apparatus we're going to talk about, the Golgi apparatus modifies the proteins made by the ribosomes coded from the nucleus, from the DNA, and it modifies them for secretion, hormones, enzymes, things like that. Mitochondria are your powerhouse of your cells. Those produce 95% of all the ATP of your body are produced in the mitochondria. And that's how the body gets energy to run. You have intermediate filaments used in supporting the cell, like scaffolding. You have microtubules, which basically guide these little vesicles that the Golgi apparatus produces towards the surface or towards where they need to go. It's like a railroad track. When these little vessels break off the Golgi apparatus, they're guided by these microtubules to an area of the cell where they're needed, or to the outside where they can be secreted into the bloodstream or into a lumen. Actin filaments, actin and myosin cause muscle contractions. Okay, these allow for contraction of the cell membrane and allow the cell to move. The cell membrane surrounds the cell, keeps it from just leaking out everywhere. It maintains an integrity for the cell. There's a nuclear membrane. Your nuclear membrane surrounds your nucleus. Okay, it's a double layer membrane, and it has pores in it because inside the nucleus is your DNA. Your DNA uses messenger RNA, translates into it. The messenger RNA leaves the nucleus into the cytoplasm to get the ribosomes to make proteins. So it goes through these pores. The nucleus is the center, is in the cell, surrounded, it contains your DNA, your RNA. Okay, your nucleolus is a dark spot. It's a dark spot inside the nucleus. And that's where your ribosomal RNA is. It produces your ribosomes that stick to your endoplasmic reticulum to make proteins. Your rough ER, or rough endoplasmic reticulum, is where all your proteins are made. Okay, they're Messenger RNA secrete out of the nucleus, goes through the nuclear pores, binds to the ribosomes on the rough endoplasmic reticulum, and makes proteins. Remember, there's primary, secondary, tertiary, and quaternary as we're developing our proteins with amino acids. Then the smooth endoplasmic reticulum. So rough endoplasmic reticulum has ribosomes. It has a little dots on it. That's why it's called rough. Smooth endoplasmic reticulum has no ribosomes. Makes your lipids, your cholesterol, your steroids, things like that are produced there. Like Glycocalyx, those are the proteins that embed into the cell membrane. Those are what communicate between the cells and allows things to pass between the cell membrane. Okay. The cell wall is in a bacteria cells, not in animal cells. Cell wall prevents disruption of the cell due to osmotic pressure, the formation of osmotic pressure with different solute concentrations. Whereas in animal cells, we have to regulate that. Centrioles are little groups at the end of the poles of each cells, and they grip the chromosomes when they duplicate and pull them toward each other when the cell divides. Chloroplast is found in plant cells. That is their material that allows sunlight to be turned into energy. It allows carbon dioxide and water to form glucose and oxygen. And flagellum we talked about, those little tails in the cell that allow propulsion and movement of the cell. Okay. So those are your basic functions of your eukaryotic cell. Okay. So eukaryotic cells, there's an external structures like your cell wall or cell membrane. You have the boundary of the cell, which allows you to have certain things in the cell and outside the cell so they don't mix. Okay, your cell doesn't leak out materials that it needs to function with. The organelles and everything else stay within the cell membrane. Organelles and the components are still within that cell membrane. There's a boundary. External structures like calyx, that is the protein that sticks on your cell membrane. This pen's not working very well. Okay, and that allows cells to connect with another. There's two types. There's capsules we talked about, and there's slime. Remember the two types? One was the capsule, which is it all over, and the slime kind of oozed out over the cell. Last chat, we talked about that. Boundary of the cell is a cell wall and a cell membrane. Those would hold your cells together, different from the external cellular environment. Okay. Organelles within the cell membrane. You have the matrix. Okay, the cytoplasmic matrix is the matrix within the cell. This pen doesn't work. Okay. The matrix is what makes up the cytoplasm and provides the media for the organelles to be suspended in. You have the nucleus, which holds your genetic material. You have your organelles, which mitochondria, chloroplast, endoplasmic reticulum, rough endoplasmic, smooth endoplasmic reticulum, Golgi apparatus, all the lysosomes, all the different organelles inside the cell. Locomotor organelles, okay, those allow flagella, allow the cell to move. Ribosomes are produced, and those are produced proteins that are transmitted the information from your DNA. And the cytoskeleton is like the scaffolding that makes up the cell. This is its shape. Nucleus is a double layer membrane. It's an envelope that surrounds the nuclear material. Inside the nuclear material is your DNA. The nucleolus is the dark part of the nucleus. That is where the ribosomal or rRNA is found. And that's where ribosomes are produced, the large subunit and small subunit, and sent down into the endoplasmic reticulum. And your chromosomes are found in the nucleus, your genetic material, your DNA. Any questions so far? I know we're going pretty quick. Stuff you probably had already in biology.
No questions.
Perfect. Okay, more of the organelles, your endoplasmic reticulum. And there's two types. There's rough endoplasmic reticulum, and there's smooth endoplasmic reticulum. You need to know the difference. Rough endoplasmic reticulum has ribosomes attached to it. It produces proteins. Your enzymes are all proteins. Different hormones are all proteins that are produced by the rough endoplasmic reticulum, by the ribosomes. The smooth endoplasmic reticulum has no ribosome. That's what's considered smooth, has all those bumps. Okay, so the smooth endoplasmic reticulum makes things like lipids, steroids, cholesterol, that kind of fun stuff. Golgi apparatus, the Golgi complex, okay, helps to modify your proteins. And mitochondria Golgi mitochondria help modify the protein. The mitochondria are your energy. Mitochondria produce ATP. Like I said, 95% of the body's ATP are produced in the mitochondria. Chloroplasts found in plant cells, the green color that allows the plant cell to absorb sunlight and turn it into energy. And that energy can be used to create glucose and oxygen from carbon dioxide and water. There's different organelles that propel the eukaryotic cell. The flagella is the end, that's the large area. The cilia are very small ones that kind of go around the whole inside, and they move on the edges and allow the cell to move through an environment. The cytoskeleton, which is the scaffold in the cell, which holds it together and allows different vesicles to go to different areas. Those are your microtubules and your microfilaments. Okay. We talked about the flagella last time, the three different parts of it. Okay. It's a long sheath cylinder, and it contains microtubules, what's called a 9+2 pattern. It occurs in doublets, okay, of these cylinders, and there's 9 pairs going around the outside, and there's 2 single ones in the center. So it's called a 9+2 arrangement that give it the ability to propel. The next slide will show you what I'm talking about. Okay, it's carried by an extension of the cell membrane. Now the cell membrane doesn't stop and the flagella goes out of it, but the cell membrane continues around the flagellum. Okay, and it's 10 times thicker than the prokaryote. So the prokaryote, remember, these are the ones that no nucleus. They're the precursors to the eukaryotic cells. So the flagellum on eukaryotes are much, much thicker, 10 times that of your prokaryotic cells. And this is a 9+2 I was talking about. These are your outer, these are your duets. See the 2 of them here? There's two. Not pencil right. 2. Gosh, can't get this pencil to right. There's 2. And then in the center, there's 2 single ones. So this is called a 9+2 pattern. There's 9 doubles. If you count all those that are around the center, there's 9 of them. And there's 2 individual ones in the center. That's your 9+2 pattern for your flagella. Okay, those are your fibers. You can see the microtubules that support the flagellum here. And the cilium are the small ones on the outside. Microtubules are on the inside. And the cell membrane does not stop here, but it goes around, completely surrounds the flagellum, and it keeps going around the rest of the cell. The flagellum, you can see how it whips back and forth, okay, like a snake-like pattern, and propels the cell through the medium. Or it can go make itself up, and it can kind of spin itself around, lashes or grabs at the substrate, and then pulls itself. So different patterns it can make. The cilia are like the flagellum, but they're very, very small. Okay? They're a similar structure to the flagellum, but they're shorter and more numerous. There's a whole bunch more and they're tinier. They're found only on single groups of protozoa and certain animal cells. They function in both motility by moving it, feeding, and filtering. Okay? You got to remember, this is your cell. And we have all these little things here. Okay? If they beat in one direction, it can move. If they beat in one direction, it can move the fluid around here, providing more nutrients to come off. Okay? And again, the surface area is tremendous. If you have a smooth surface like this or a rough surface like this, look at all the surface area you've increased. Okay, with your cilia. So it helps in feeding because more stuff can be absorbed, and in filtering, it can filter more of an area. Okay, so it increases your surface area, so it allows for filtering and feeding, and it helps in mobility as it moves. As you can see all the cilia right here. These are your cilia. All these little hair-like structures that surround the entire cell. You can see if they all push in one direction, they can propel the cell. If they come up and then they can recover by pushing back to where they originally were. And they can propel the cell in one direction. So I call it glycocalyx. We talked about that last time. It's your boundary that comes in direct contact with the environment. They're usually composed of your polysaccharides. Remember, your saccharides are your sugars. Polysaccharides are 3 or more. You got monosaccharides, disaccharides, and polysaccharides. Okay? They appear as a network of fibers. They have a slime layer or a capsule layer, which you talked about. They function in adherence, so they bind the cell to another cell or to another surface. Okay? They allow for protection by preventing certain things from going into the cell. Okay? And they're a single reception. In other words, they allow certain things in. They have receptors that will recognize it and allow things to pass into the cell. So beneath the glycocalyx, fungi and most algae have a thick, rigid cell wall, not in animal cells, but in fungi and most algae, there's a rigid cell wall, and in plant cells. Protozoa and a few algae, and all animal cells lack a cell wall and have only a cell membrane. And again, the important part is cells that lack a cell wall are susceptible to osmotic pressure changes, which is really important for when we give IV fluids to a patient. All right, the cell wall in plant cells, some algae cells. They're rigid, provide structural support, and they shape the cell. Okay. Fungi. Fungi have a thick inner layer of polysaccharide fibers composed of chitin and cellulose and a thin layer of mixed glycans. So chitin is found in the cell wall. Algae, varying chemical composition, substances commonly found in cellulose, pectin, mannins, silicon dioxide, and calcium carbonate. Okay, this was found in making up the cell wall in algae. The cell membrane is a bilayer of phospholipids we talked about. The hydrophobic fatty acid chains are on the inside. The hydrophilic phosphate heads are on the outside. So it forms a phosphate bilayer phospholipid cell membrane. Sterols confirm stability. Cholesterol, things like that, wedge into the cell membrane and give it its stability. Serve as a selective permeable barrier and transport certain things across that cell membrane. It helps to transmit nutrients in and helps to transmit waste out. Eukaryotic cells also contain membrane-bound organelles that account to 60 to 80% of their volume. Remember, prokaryotic cells do not contain a nucleus and do not contain organelles. Eukaryotic cells contain organelles, and some are membrane-bound, like mitochondria, endoplasmic reticulum, Golgi apparatus. Those are membrane-bound organelles, and they account for about 60 to 80% of the volume of the cell. Okay, so what part of the eukaryotic cell is responsible for contacting the outside environment and signaling between cells? Would it be the flagella, the cell wall, the glycocalyx, or the cell membrane?
Is it the flagella?
Now, the flagella allows for movement.
Oh, is it glycolic?
Yep, it'd be glycalyx. That's the one that binds your cell membrane and allows cells to connect to one another, and it's signaling between them and allows you to connect with the outside membrane. They're actually found in the cell membrane. It allows it to transfer things. So it should be C, and it is. Okay. This is a summary which just went over. Okay. Nucleus, genetic center of the cell. Okay. Nucleolus, this is with your rRNA. Dark spot in your nucleus is your ribosomal RNA. This is where we make ribosomes. Endoplasmic reticulum transports material, lipid synthesis. In the smooth endoplasmic reticulum, rough endoplasmic is protein synthesis. Golgi apparatus modifies the proteins that are made in the endoplasmic reticulum. Okay, lysis digest pathogens, things that get into the cell. Vacuoles are storage where they digest things and break it down, and they store these for later use. These so you don't waste amino acids, things that have been broken down from anything else in the cell. Mitochondria produce energy. Use the Krebs cycle, electron transport chain, oxidative phosphorylation. Chloroplasts convert chemical energy, convert sun energy, solar energy, into sugar, which is glucose and oxygen, through photosynthesis. Ribosomes make proteins. Okay, they make protein, and they're located on the rough endoplasmic reticulum. That's what makes it rough. Cytoskeleton is your scaffolding that holds the cell together on the inside. The nucleus. Nucleus is where the DNA is, right? It's a compact sphere, most prominent organelle of the eukaryotic cell, easy to see. Composed of two parallel membranes. Remember, it's a double membrane. There's two compound, two parallel membranes. There's a narrow space in between both those membranes, and it's perforated with pores. Pores allow the RNA that's been translated from the DNA to go out of this nucleus into the cytoplasm, where it can be read and interpreted to make proteins. Nucleolus is a dark area inside the nucleus. Okay, it's where ribosomal RNA synthesis occurs and ribosomes are assembled. The large subunit, the small subunit. Remember that little staple action that it does? That's where they're made. Here you can see the nucleolus. It's 2 layers thick. You can see a space in between there. You can see it 2 layers thick. You can see the pore right here, nuclear pore. So RNA can get out. This is the nucleolus. This is a dark area where the ribosomal RNA is. This is your chromatin. This is your DNA. And all these little patterns here, this is your endoplasmic reticulum. So your RNA comes from your chromosomes, comes out into the endoplasmic reticulum, where it's bound to ribosomes and makes protein. Okay. Again, you see a nuclear pore between the double layers, outer layer, inner layer of your nucleus. And there's pores in between. This dark center is not your DNA. Most students get that wrong. The dark center inside the nucleus is your ribosomal RNA. That's the nucleolus. Mitosis. Mitosis is how a cell divides.
I have a question.
Yeah.
You said it's not the nucleus, it's the nucleolus, is the is the RNA?
Yeah, this nucleolus here, this is ribosomal RNA. This is the RNA that makes ribosomes. And then transfer into the cytoplasm. Your DNA is out to the edges here. Your chromatid. Because most of the time the cells function, the DNA is not condensing the chromosomes. It only condenses the chromosomes for replication. Your DNA is all spread out, not dark or anything like that, because it's being used. You're transcribing it. You're transcribing it. Okay? It's binding RNA. It's using its genetic code, so it's not condensed. The only time DNA condenses, it condenses into chromosomes for replication, for mitosis. So the dark spot inside the nucleus is your nucleolus. That is not your DNA. That is the rRNA. And most students on the nursing exams get that wrong. They say, oh, that's the chromosomes. No, the time it spends in chromosomes is very small because that's only when it divides. The rest of the time, the cell's undergoing all its replication, all its proteins, and everything. The genetic material is all spread out, so we can read it. Does that make sense?
Yeah, because the ribonucleic acid is only responsible for transmitting like information from the DNA. Like it's not actually it just replicates the code. It doesn't actually make the genetic material like DNA does, right?
Correct. DNA is the genetic code. The DNA is a double chain, right? Remember, it's like a ladder that's twisted. Okay, so you got 2 strands. These strands open up because there's hydrogen bonding between it, so they open up. And these areas, like it's adenine, guanine, cytosine, whatever it is, these areas allow messenger RNA to bind to this using the opposite bases, like adenine bonds with thymine, cytosine bonds with guanine, but in RNA, it's uracil bonds with adenine. And then that messenger RNA duplicates this code, comes out through the nuclear pores into the cytoplasm, and that's what makes your protein. So that's your genetic material. Okay, this is your ribosomal RNA that makes ribosomes for protein synthesis. Does that confuse you or make sense?
No, that makes sense. Yeah.
Okay, perfect. So that when it undergoes mitosis, the only thing you got to remember for your nursing boards, remember the term PMAT. Okay, that's the mnemonic for it. Prophase, metaphase, anaphase, telophase. That's your steps of mitosis. Okay, so prophase, you start to form a chromosomes. See, at the beginning here, this is interphase. This is your beginning, your resting stage. You see your DNA is all spread out. It's making proteins. It's transcribing into messenger RNA. Now it starts to condense together. This is prophase. You start to form chromosomes. The DNA starts to combine, coil, wrap itself around histones, which are little proteins, and form chromosomes. Okay, that's prophase. Metaphase, the chromosomes line up in the middle. So prophase, you start to make chromosomes. They start to condense. Metaphase, okay, metaphase, they all line up in the center. Anaphase, they start to pull to the different poles. They pull from one end to the other, leaving in the center. And then telophase, you start to get separation. You can see the cleavage here, where this cleavage starts to form. You start to get separation into two daughter cells. Okay, then it goes back into the daughter cells, go back to the resting phase, make proteins and everything else for the cell. And then when they want to get ready to start, they start mitosis again. So prophase, metaphase, anaphase, telophase, PMAP. Prophase, chromosomes start to condense. Metaphase, they line up in the center. Anaphase, they start to pull apart. Telophase, the cells start to split into two. That's mitosis in a nutshell. Endoplasmic reticulum, two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay, the rough endoplasmic reticulum connects with the nuclear envelope, extends into a network through the cytoplasm, and is rough due to the attachment of ribosomes. This is where protein synthesis occurs. Okay. It goes into the endoplasmic reticulum and gets packaged for transport. And that's the first step in the secretory pathway for proteins that are formed. The smooth endoplasmic reticulum does not connect with the nuclear envelope. It's a closed tubular network without ribosomes. There are no ribosomes in SCR. Functions in nutrient processing, synthesis of cholesterol, lipids, and then the storage of lipids also. This is rough endoplasmic reticulum. You can see all the little blue bumps on it. That's your ribosomes. All these little things are ribosomes. They're composed of cisterns, which are caverns, as this folds back and forth, and it has a lumen, which is an opening inside. The endoplasmic reticulum breaks off into these little transport vesicles. These transport vesicles go to the Golgi apparatus. Once it goes in the Golgi apparatus, it grinds through that and gets more refined, and then it gets compacted into another transport vesicle. So it goes from the nucleus into this rough endoplasmic reticulum, and you make your protein. The protein goes through the endoplasmic reticulum, all the channels, and gets modified. It gets secreted into a little taxicab, a little vesicle, goes out, goes to the Golgi apparatus. That little vesicle binds to the Golgi apparatus. It fuses with it, just like it does here, and it secretes its protein into the Golgi apparatus. The Golgi apparatus is another series of canals. It runs through there, it gets further modified. When it's finally ready to go, it goes to the end of the Golgi apparatus, and then it branches off into another secretory vesicle. Now, this vesicle does one of two things. It either goes to a part in the cell by fundus microtubules to an area where it's needed, or it goes to the cell membrane, where it is engulfed and is spit out into the bloodstream, lymphatic system, into the digestive tract, wherever that cell is. It's excreted from the cell into a lumen or outside the skin or something like that. Okay, but it's excreted from the cells. So it goes from nucleus, endoplasmic reticulum, Golgi apparatus, secretory vesicle gets excreted. Okay. This is your messenger RNA right here. Your strand that was made from the DNA went through the nuclear pore. Okay. You got your small subunit and your large subunit. It's like a little stapler. It just adds every 3 codes. It adds 1 amino acid, and this amino acid chain grows. And as this chain grows, this is what's called your primary. There's primary, secondary, tertiary, and quaternary proteins. This is your primary protein. This is just your chain of amino acids. The secondary one was, remember, beta pleated sheets or alpha helix. The tertiary one was your 3-dimensional shape. The quaternary one was the bringing together of 2 or more chains in 3 dimensions to make a functional protein. Golgi apparatus we talked about modifies, stores, and packages the proteins that are made in the rough endoplasmic reticulum. Contains these flattened sacs called cisternae And it's like a stack of pancakes, is basically what it looks like. It's smaller and smaller and smaller as it goes up. Okay, this is your Golgi apparatus right here. These are the stacks that it makes. And then the secretory vesicles, the ones that come out, either go to a part of the cell that needs a protein rating or gets excreted outside the cell into the extracellular fluid. So synthesis is nucleus, rough endoplasmic reticulum, Golgi apparatus, vesicles, and secretion. Okay, the nucleolus provides ribosomes. Remember, this is your ribosomal RNA, rRNA. And that's what makes your ribosomes, it codes for it. That's your nucleolus. 80% of the people that take the N class, I guarantee you, they're going to say the nucleolus and they're going to check off the box that says the word DNA is located. And it's not true. Okay? DNA is located as chromatin throughout the nucleus. When it condenses, then it becomes chromosomes. The nucleolus is a dark area of the nucleus where the ribosomal RNA is that are producing ribosomes that are transmitted into the endoplasmic reticulum to make rough endoplasmic reticulum to process protein synthesis. Does that make sense? Don't miss that many nursing exams, okay?
Yes.
Okay, cool. The transport vesicles from the endoplasmic reticulum go to the Golgi apparatus, okay? And where they're further modified and matured for whatever their function is going to be. Condensing vesicles after the Golgi apparatus then transmit those secretory vesicles to either outside or into a lumen like a blood vessel or outside the skin, okay, outside the body, or it goes in the body, inside the cell to an area that needs it. Those vesicles can go to two different places. So here it is. Okay, this is your nucleolus. And your nucleus is a dark spot, and it makes ribosomes. And here these little ribosomes are coming in, and they're binding to the reticulum where they're going to make rough ER. Okay? And from the once the messenger RNA comes out of the nucleus here into the rough reticulum, it goes through it, through the different matrices, and comes out as this transport vesicle. This transport vesicle fuses with the Golgi apparatus, and then it works its way through the Golgi apparatus, where it creates a condensing vesicle. Condensing vesicle can go to different parts of the cell where it's needed, or can fuse with the cell membrane and kick out the secretion by exocytosis. Get rid of hormones, make your body act in certain ways, etc. That's how it works. Lysosomes. Lysosomes are these vesicles that eat things that are foreign to the cell. Okay, pathogens, things like that. They contain enzymes that digest, originate from the Golgi apparatus. Now they can break down good parts of your cell too. I mean, things wear out. So some of them go to the lysosome, they break down the existing part of the cell where they're worn out, and then they recycle the components of it. Okay? So it's not just for pathogens. It's involved in extracellular digestion. Intracellular digestion. What's in the cell, it digests and breaks it down. Okay? Protection against invading microbes, different pathogens break down. Vacuoles are membrane cells containing particles to be digested, excreted, or stored. Phagosomes are vacuoles that emerge with lysosomes and break down pathogens or other parts of the cell that have been worn out. And this shows you an example of synthesis and the transport of where it goes. Food particles come in, they go into the food vacuole. Okay, and they fuse with lysosomes, which breaks them down, can get digestion, and they can use these as nutrients. Okay. Okay, the mitochondria. Mitochondria produce ATP. That's their function. Remember, mitochondria produce ATP. They're spherical organisms. They're like rod-shaped. Okay, they have an outer membrane and an inner membrane. They're double membranes, the mitochondria. Okay, and the folds in them are called cristae. Cristae membranes hold enzymes and electron carriers of aerobic respiration. Aerobic respiration means in the presence of oxygen. In the presence of oxygen, it runs through the Krebs cycle, and it breaks down glucose into pyruvate, and then pyruvate goes through the Krebs cycle and produces large amounts of ATP. They divide independently of the cell to make more mitochondria. They don't divide with the cell. The DNA and the prokaryotic ribosomes are contained in spaces around the cristae are called a matrix. It's called the matrix of the ribosomes. Okay. You can see the matrix, which is this inner portion here. These are the cristae. The cristae are all little folds. The matrix is the inner portion. These are the ribosomes that are in the mitochondria. There's mitochondrial DNA, which has been used recently in a lot of forensic cases because a lot of the DNA is not usable. Mitochondrial DNA is more stable. There's not as much of it, but it's a lot more stable than hereditary DNA. You have an inner membrane and an outer membrane. And this is what a mitochondria looks like. These are all the ribosomes in rough endoplasmic reticulum. Chloroplasts. Chloroplasts are found in plant cells, not in animal cells. They convert the energy of sunlight into chemical energy through photosynthesis. Okay, that's your chloroplast. It's found in algae and plant cells. The outer membrane covers inner membrane folded into sacs called thylakoids. These thylakoids are stacked like a stack of coins. You're a gambler in Las Vegas or casinos, you have your stack of coins. Those are like your thylakoids. And the stacked one is called a grana. Okay, they're just stacks of discs. Okay, those carry your pigments and your chlorophyll, which allow photosynthesis to occur. They're the primary producers of organic nutrients for all other organisms. They're the ones that produce glucose. Okay, from CO2 and water. There's your stack, your granule. Your thylakoids. Each one of these is a thylakoid, and each stack is a granule. They're like stacks of poker chips. Okay. The stroma is your matrix between each one. These are your ribosomes that are in there. And the chloroplast is this double membrane here. This is your chloroplast, which contains thylakoids and the granums. All right, in the ribosomes. Okay, they're composed of ribosomal RNA, which produce them, and proteins. Scattered in the cytoplasm, and they're associated with rough endoplasmic reticulum. So they can be associated with endoplasmic reticulum, and they can also be free-flowing throughout the cytoplasm. They don't have to be bound to the rough endoplasmic reticulum. That's what most of them are. They're larger than the prokaryotic ribosomes from prokaryotic cells, and they function is protein synthesis. That's what ribosomes do. They make protein by stapling a chain of amino acids together. Again, you can see the endoplasmic reticulum. See the ribosomes, little blue dots that bind to it. The lumen is the opening of the space in where the protein are going to form. And they function by reading messenger RNA, and they act like little staple guns. And they staple and make this chain longer, longer, and longer until it reaches the correct length it's supposed to be. The cytoskeleton is the inside of the cell. That's a flexible framework of the microfilaments and microtubules that form your network throughout your cytoplasm. It's involved in the organelles anchoring. And so it keeps them in place. Movement of the cytoplasm. Amoeboid motion, which means your cell moves by changing its shape. Transport and structural support. So they basically support the inside of the cell and support its shape. Okay, remember, animal cells have no cell wall, so this is all controlled by these microfilaments, microtubules. These guide your proteins that have been made from the Golgi apparatus, and they guide them along like a freeway or like a little pathway, either to the outside, to the cell membrane where they can be excreted, or to other parts of the cell where they can be used. And these are what the strands look like. These are the microtubules. And you can see these tubular subunits here that run throughout the cell, and these control the shape of the cell. They control the network in the cell, how things move through the cell. You have actin filaments make up these here. These are part of your microfilaments. Remember, actin and myosin are contractile elements. So they allow the inside shell to contract, and you get amoeboid movements or motions as the cell moves. Okay, so prokaryotes eukaryotes, and viruses. This just gives you a little bit of what they have. Nucleic acids, do they have a true nucleus? Only, they all have nucleic acids, all three of them. A true nucleus is only found in eukaryotic cells. A nuclear envelope is only in eukaryotic cells. Turn that off. There you go. All right, nucleoid is prokaryotic cells is an area. The nucleoid is an area where the genetic material conglomerate. There's no membrane around it. It's just a mass of genetic material. It's called a nucleoid, like a nucleus, but it does not have a membrane. Reproduction of eukaryotic cells, mitosis. Okay, prokaryotic cells, production of sex cells could be prokaryotic. That's how eukaryotic cells divide. Binary fission, it's a breaking apart, just one thing breaks into two. That's your prokaryotes and your eukaryotes. Biosynthesis, all the different areas. You can see the viruses don't have any of it. Viruses lack all the material to replicate themselves. They have to use their host cell to replicate. They cannot do it on. Different types of respiration, photosynthesis. Okay. Cell wall, you notice here the viruses have a capsid, which is just a protein coat around it, not a true cell membrane or cell wall. Okay. But both prokaryotic and eukaryotic cells have can have cell walls and cell membranes. And where else we have? Perfect. So looking at that table, it positive means you represent characteristic. The minus means most lack that characteristic. Viruses cannot participate in metabolic or genetic activity outside their host cell. So if a virus is not in a host cell, it is non-functional. Can't undergo, it can't replicate itself. Okay, and it can't grow, can't do anything. It has to use the host machinery once it infects a cell to replicate itself or to grow. Prokaryotic types are very different than eukaryotics. They're much smaller, much larger to exist. We'll check on that. So the eukaryotic organelle that's responsible for transporting vesicles inside the cell is what? What is responsible for transporting vesicles inside the cell?
Is it C, rough endoplasmic?
That's what makes them.
I mean smooth endoplasmic.
That's what makes your Golgi apparatus. That's what makes your steroids and your
Oh, Golgi.
Golgi. That's the one that takes those vesicles from the reticulum and then modifies it and then releases it into another vesicle that tells it where it's going to go. So that's what's responsible for transporting these vesicles inside the cell, where it's going to go, where it's going to be exocytosis, excreted from the cell, or to another part of the cell. So the Golgi apparatus control where the vesicles are going to go inside the cell. A family tree, basically, of eukaryotes, where they branch into the red algae, the chlorophytes, brown algae, diatoms, okay? Branch in there, and you can see the branch that they form of all the different domain groups. Okay, and so you have one eukaryote branches into all those different areas. Okay, so eukaryotic microorganisms, you have fungi, you have algae, protozoa, and parasitic worms. Those are microorganisms broken down. The kingdom of fungi. The majority of fungi are unicellular or colonial. Okay? A few have cellular specializations. There's 100,000 species of fungi, and they're divided into two groups. There's macroscopic fungi, and there's microscopic fungi. Okay? Macroscopic fungi you can see with the naked eye. Microscopic fungi, you need a microscope to see it. Your macroscopic, your mushrooms, puffballs, gill fungi. Microscopic fungi have two morphologies. They're your hyphae and your yeast. Hyphae are long filament fungi or molds. Your yeast are round, ovoid shaped. They're produced by asexual reproduction. Okay. That's your breakdown of fungi. Right? And you know all what fungus looks like. The hyphae are long thread-like cells, make up bodies of filamentous fungi or molds. Some present various textures like mycelia, another array of different colors. Okay. It's macroscopically due to spores that it forms. Okay. This is a hyphae. You can see all the different filaments basically. It's called septum as you look at the microscope. And yeasts. See, yeasts are round, they're not long and rod shaped. Okay, yeast are round and oval shaped. Okay, it's made and it occurs by asexual reproduction. A piece buds off and forms a second one. Presents a cell wall and lacks locomotor organelles. So there are no cilia and there are no flagella in yeast. They don't move around. It grows swellings on its surface called these little buds, okay, which then separate into cells. And that's asexual reproduction.
So that's what like a yeast infection is?
Yep, exactly what a yeast infection is. Okay. And it grows into cells. You can get a part budded off and form two cells, another part is budded off, and it can form this yeast chain. That's the stuff that forms the white lines, like in the yeast infection in your mouth or yeast infection somewhere else. You see these white chains being formed. And that's what the yeast look like under a microscope. Fungi. All fungi are heterotrophic. Okay? Majority are harmless safe probes living off dead plants and animals. But some are parasites living on the tissue of other organisms, but none are obligate. Okay? There are mycoses. They're fungal. Mycoses are called fungal infections. They're called mycoses. Extremely widespread distribution in many habitats. They grow in a lot of areas. Anything moist, wet areas, they will grow in. You can see how they grow in these different shapes, different fungal infections, and they'll spread out into different colonies. Okay, fungal infections, a lot of occur on people's on the feet. You get a lot of fungal infections on your foot, athlete's feet, etc. And this is what they look like. This is a hyphae being together in your linear cells.
Do you get that from like wearing like certain shoes or like taking like showers in places that are like dirty?
Yes, you get it from showers from other people. It can be transmitted by contact. And different types of shoes that allow your feet to sweat, and they can't get rid of that, they'll go into fungal infections. Yep, exactly. Now, the microscopic fungi is the ones you can't see. Okay. There's filamentous fungi, which is molds. There's cottony, hairy, or velvety texture. They're woven, intertwining, massive hyphae. Remember those long lines that we saw? Okay. And those are called mycelium. They may be divided by cross walls, which are called septa. Remember before that, we can show you, there's the septa here. Here's your cross walls. This is a septa. You can see these long forms of it. So the mycelium is the chain that's developing from the fungi. Those are your hyphae, and then divided by crossover to your septa dividing. You have vegetated hyphae, which is digestive and absorbs nutrients, and you have reproductive hyphae, which produce spores, so they reproduce. And with yeast, okay, some form a pseudohypha, which is a chain of yeast form, buds remain attached in a row. Remember those when they bud off, they form a row and row and row? That's like a pseudohypha. It looks like a hyphae because it's all connected, but they're just buds of the cell reproducing. So, and just don't break apart. Because it's from that, it's not a true hyphae like that of mold. Molds are individual cells that link together. These pseudohyphae or yeast are the little buds that break off, and they just don't completely break off with their individual cells. You can see how it forms. This is the septa here, and they form into groups. And then the buds that form, this little bud right here, and they just don't separate. So it's like a pseudo hyphae, and this is a actual hyphae. Where they form septa, you see all the dividing points are the septa right here. My pen will work. Right there, right there. There's a little septa. Okay, fungal reproduction. The reproduction of fungus is usually through spores. Okay. It's formed out of what's called reproductive hyphae. Okay. The hyphae form on the surface of it. Okay, they go into sporangia, which is a sphere that contains the spores. The sphere ruptures, and these spores are released. Spores are released, go through a germ tube, a hyphae, surface hyphae, and then form the cycle all over until more spores are formed. So fungi reproduce by spores. Their spores are very difficult to kill. Sporangiospores are formed by successive cleavages within a sac-like head called a sporangium. That's what forms all these little spores. They initially enclosed but released when the sporangium ruptures. All these spores are released into the external environment. In conidia, those are free spores that are not enclosed by a spore-bearing sac. They just formed on their own. Okay. So here's your sporangiospore. All these little spheres are spores inside of it. When that sac ruptures, all these little spores are released. All right. Sexual spore formation. Spores are formed following fusion of two different strains and formation of sexual structures. There's basically male and female. They come together, you get fusion, and then you get sexual reproduction, two different genetic materials now. Zygospores, ascospores, and bacillospores are formed. Sexual spores, spore-forming structures, are on basis for classification. That's how you can classify different things by what forms them and how they're formed. These are zygospores. Zygospores like mold on bread. You get two hyphae coming together, basically a male and a female, or plus and a minus, either way. And genetic material is exchanged when these start to form. They combine the genetic material, germination, and then they get spores that are formed from the union of the two hyphae. So that's what they're called, zygospores. Ascospores. Okay. Ascospores have a male and a female portion. Okay. They fuse together, they form this called these fruiting bodies, as this genetic material is mixed between the two as they're formed. And then they release these spores that are a combination of the two types of genetic material. So that's ascospores, how they're formed. And bacilliospores. What? Is there a question?
Oh, I was just saying that ascospores are formed on, they're fruiting, like where do
Yeah,
you find?
fruiting bodies. These fruiting bodies form together, the two combined, because these are the fruiting bodies right here at the top. So once these are formed, they're formed by genetic material of the two types as they've merged. Okay, they've merged here and formed this. And as these bodies open up, and these spores are released. And each spore now is a mixture of the genetic material of the two different types.
Okay,
Okay.
thank you.
Sure, no problem. Bacilliospores. Okay, this is how your mushrooms are formed. Those are the filaments at the end of the mushrooms. This is the above ground structure and the below ground structures. You can see that the formation, okay, as they come across there. The bacilliospores vary in genetic makeup. Okay. Then they'll come together. In other words, there'll be different types of basically a plus and a minus. Okay. The strains will start to develop, and then the strains will fuse. When they fuse, it's like a male and a female or positive minus coming together, and then genetic material is combined. Okay. These form mycelium, and then the mushrooms start to form. And then you got these places of caps right here on the top, and these parts rupture, and the inner parts come out of that, and these form these different types of mixed genetic material now. And that's formation by bacillospores. That's how mushrooms reproduce. Okay, classification of fungi. Okay. They're divided into several phyla based upon the type of sexual reproduction. There's phylum Zygomycota, Ascomycota, Basidiomycota, Clitriotomycota. Okay. If it's asexual, form is called imperfect because they're not mixing genetic material. So Zygomycota is zygospheres form, like we just showed the digospheres, mostly sporangospheres and some conidia. The Ascomycota, ascospores and conidia. Basidiomycota, the spores like the mushrooms. And Clitriotomycota flagellated spores, ones that actually have flagellum propelled. There's different classifications for fungi. We identify different fungi based on a specific media that we put it on. Okay, macroscopic and microscopic observation, the asexual spore forming structures and spores, okay, the type of hyphae that it forms, the colonies that it forms, the different pigmentations that form, the physiological characteristics of it, and the genetic makeup. Use all those classifications to help identify different types of fungi. Okay. So zygomycota. Okay. Those are sexual spores, zygospores. Okay. The asexual spores are sporangiospores. Hyphae are usually non-septated. In other words, they don't have the septa dividing between them. If septa, the septa are complete, dividing each off, branch off. Most species are free-living saprobes. Some are animal parasites. Saprobes is one of the divisions we had on the, as we divided the different groupings. Okay. Can be obnoxious, contaminants in the laboratory, causing food spoilage agents, destructive to crops. Okay. Examples of common molds, okay, are black red mold, Rhizopus, Mucor, Obsidius Cernella, and Circinella. Those are all different types of common molds. Okay. This is a Syncephalustrum. Okay. You can have the soil and plants. You can see the mold. This is Circinella. Okay. Known for its curved sporangiophores. See how they curve around here, where they're going to bud off and release their spores. Okay. Ascomycota. Okay. They have sexual spores, produce ascospores in asci. They have asexual spores. Okay. Many types of conidia form the tip of conidiophores, and hyphae with porous septa, openings between the septa. This is the largest phylum, vary from macroscopic mushrooms to microscopic molds and yeasts. This is your largest phylum. Other representative method of that phylum are Penicillium, it's a source of antibiotics, Aspergillus, commonly airborne mold, involving respiratory infections, Candida pseudohyphae, a yeast infection, yeast that's used in making bread and beer. Many human and plant pathogens, like pneumocystis, okay, as a pathogen of AIDS patients. Includes many human and plant pathogens, like pneumocystis, Jovovecki, the same as the pathogen of, again, the AIDS patients. Histoplasma is Ohio Valley fever. Trichophyton is one thing of ringworm, common name for certain fungal skin infections that often go like a ring pad magazine. Coccidioides immitis is a cause of valley fever. Candida albicans is a cause of yeast infections. Decobotritis is a toxic mold. So there's different types representatives of this group. Aspergillus fumigus, okay, displays it's like a flowery appearance of these spores that are going to separate. And these are the different buds that you saw before, as these buds and stick together. Scanning electron microscope of Candida pseudohyphae. Remember, it's pseudohyphae because it doesn't have real septa dividing it, and they're not growing as one piece. They're the buds that form off that branch. They don't completely separate, and it forms this fake or chain, basically, where the buds just don't separate. That's what's called pseudohyphae. Okay. Basidomycota. Okay. Microscopic forms such as mushrooms and puffballs. Okay, includes a number of microscopic plant pathogens called rusts and smuts that attack and destroy major crops. Okay, there's an extensive impact on our agriculture and global food production. All right, continuing on that, they can have sexual reproduction by the basidia, just like with the mushrooms we talked about, and the Vispidiospores, or if they do asexual reproduction through spores of Canidia. You have incomplete septa hyphae. Okay. Remember, they're the buds that form, so they're completely separated by the septa. Some plant parasites. And there's one human pathogen that attacks humans. Fleshy, fruity bodies we talked about are common. As those fruity bodies open up, those spores are released. An example of this group is mushrooms. Okay, mushrooms, puffballs, bracket fungi, and plant pathogens, rusts and smuts, which affect the crops. The one human pathogen called Cryptococcus neoformans causes invasive systemic infections, several organs, including the skin, the brain, and the lungs. Okay, close up of the fly argonic mushrooms. You can see the stalk that forms, what's here, and underneath that are your septa underneath it, where all your spores are going to form. They come out through reproduction. This is Cryptococcus neoformans. This is one that is infectious to humans. Okay, it causes lung infections, brain infections, skin infections. Okay, it's a yeast that causes cryptococcosis. There's capsules around every cell. Those ones that form the infections. Chytridomycota, members of family unusual primitive fungi. Their cellular morphology, basically their shape, ranges from single cells to clusters and colonies. They generally don't form the hypha that we usually see, or yeast-type cells. Okay. The one thing that we characterize them by is the presence of what's called of a special flagellated spore called zoospores and gametes. Most chytrids are saprobic, free living in the soil, water, and decaying matter. There's a significant number of parasites in plants and animals and other microbes. They're not known to cause human diseases. There can be serious frog pathogens, often responsible for destroying whole populations in some habitats of frogs. Okay. Parasitic chytrid fungi. You see the diatoms. Diatoms are two of the cells fused together. Okay. Attacked by flagellated chytrid, you can see the flagellum. Right here. Okay. Flagellated chytrid and destroy their host cells. So these invade the host, they start to separate and destroy it, but that's how they reproduce. So use of host material. Fungal involvement in medicine, pathogenesis, nature, and entry can be at two levels. There can be an adverse impact, or there can be a beneficial impact. Mycoses are an adverse impact. Mycoses are allergies and the toxins that are produced. We have destruction of crops and food storages from these fungi. Fungi must also be a source of antibiotics, alcohol, organic acids, and vitamins. It decomposes dead plants and animals. It's used in making foods and in genetic studies. These are some of the major infections in humans. This is the type, and then you can see the different fungi that cause the different fungi infections, like Candida albicans causes your yeast infections in humans. Cryptococcus neoformans, where it affects your lungs, your brain, your skin. That's Cryptococcus bacteria, fungus. Okay. So different types of fungus cause different types of diseases. So fungi are generally classified according to their type of sexual spore, type asexual spore, type of hyphae, type of habitat. How do we usually classify
Type of hyphae?
by the hyphae? Perfect. By how they form. How they form their extensions and how they combine to form by reproduction. Perfect. The protists. Protists are algae, protozoa. Algae are eukaryotic cells or organisms. They're usually unicellular and can be colonial. They photosynthesize with chlorophyll. So you have chlorophyll, they produce energy using sunlight. Protozoas are unicellular eukaryotes. Okay. They lack tissues, share similarities in cell structures, nutrition, life cycle, and biochemistry. Okay. So algae are photosynthetic, microscopic forms are unicellular, macroscopic forms are colonial and multicellular. They contain chloroplast, chlorophyll. They have a cell wall and may or may not have flagellum. So this is what they look like. This is an algae. You can see the flagellum, you see the ribosomes, okay? The mitochondria, the nucleus, nucleolus. Remember, this is your nucleolus, the dark spot here. Chloroplast, your green areas that allow you to convert sunlight from photosynthesis. Golgi apparatus for processing your protein. Cytoplasm surrounds the nucleus. Cell membrane, cell wall, vacuoles. Things that we've seen before. Most are free living in fresh and marine water, like plankton. Provide the base of the food web, most aquatic habitats. Produce large portions of atmospheric oxygen. And one of their byproducts is glucose and oxygen. Dinoflagellates cause the red tides. What we see down at Point Magoo down here in the beach on the Marine Corps base, there's a red tide that comes across when the tide glows. Those are the toxins given off by the dying dinoflagellates. They give out toxins that cause food poisoning with a neurological symptom. So not fun stuff. But this is red tide. And if you watch red tide at night, it glows in the dark. It's really pretty. It's gorgeous. As each wave breaks, it becomes fluorescent and glows. Very toxic. So we don't want to sit and drink the water. They're very pretty. And see this algae here, you see green algae for photosynthesis. Okay. These are some of the different common names. There was cell wall, types of pigmentation they have in them for all the different algae. The importance of each one. This is what causes red tide. This is your dinoflagellates cause red tide. Major component of plankton is your diatoms, your golden brown algae. And continuation that it can cause is this is a food additive here. This is like red seaweeds, an emulsifier, alginate. This is your brown algae and kelp beds. Okay, so the protozoa. Protozoas are diverse group. There's about 65,000 species of protozoa. They vary in shape and they lack a cell wall. So just like animal cells, they don't have a cell membrane for protozoa. Most of them are unicellular. Most are harmless. They live in a moist habitat. Some are animal parasites and be spread by insect vectors. Remember we talked about insect vectors before? Like a mosquito that spreads malaria. All are heterotrophic. They lack chloroplast. What's that?
I have a question back to the animal parasites. Is that
Yes.
the like in the category of like Lyme's disease, how it's transmitted through like a deer?
That's an insect vector, yes. That could be spread by a different vector, yeah. But insect vector is the one spread by insects. Lyme's disease is that spread by the? I'm trying to
Like
remember what Lyme's.
It's spread by it's called a deer tick.
I don't remember the type of tick that spreads it. Is it called a deer tick?
Yeah, I think.
Okay,
it's
so that the tick could, yeah, be an insect vector. The tick gets infected and it spreads it when it bites you. It ejects toxin into your blood.
Okay, yeah. A lot of people are getting that. I've seen like on the
Yeah.
news.
Insect vectors are a very common way of transmitting diseases. Yeah, it's a Lyme disease is a very nasty disease. A lot of times they're very difficult to treat. Once the tick bites you and gets into your bloodstream, you become septic in your own blood. Can be very difficult to treat, and the treatment can last for months. It's not something easy to treat.
Yeah, it can mess with your like it can have nausea, brain, like I don't know, fatigue, all that.
Yep, definitely causes mental fatigue. Yep. I thought it was something different than name. I'll look it up though. Look up deer tick and Lyme disease. All right. On the protozoa, those are protozoas. They're all heterotrophic. They lack chloroplasts. The cytoplasm is divided into ectoplasm and endoplasm. So there's two divisions of cytoplasm in the protozoa. They engulf other microbes and organic material, and that's how they feed. They're shaped like that. So how they feed is they invaginate their cell membrane like that. And food particles travel in, then it seals them off, seals them in vacuoles, and digests them. Okay, so they feed by engulfing other microbes and organic materials. Most have flagella or cilia or pseudopods, allows them to move. They exist as trophozoites, which are mobile, okay, feeding stages. They move around to different areas to feed on them. They're made in a dormant stage when there's unfavorable conditions for growth, okay, which is called a cyst. Basically like hibernation. There's not any food around, not nutrients around, it goes into hibernation. It's called a cyst. All reproduce asexually. Mitosis or multiple fusion. Many also reproduce sexually by conjugation. So all can produce asexually just by budding off, and some can also reproduce sexually by conjugating, by fusing two protozoa together and sharing their genetic material. Okay. So this is your active feeding stage. We have different invaginations of the cell membrane. Okay. When it dries up in the moist environment, it forms this cyst wall around it. Okay. This is a mature cyst. Okay. It stays this way until the environment changes, where it gets more moisture, more nutrients. Okay. Then this cyst wall breaks open, and this cell becomes feeding again. Okay. So basically, it forms a cyst when there's not enough in the environment to support it. Okay. Unique appearance of most protozoa allows us to identify kind by levels of genus and species. By microscopic morphology. Okay. Identification, we use the size and shape of the cell. We use the type, number, and distribution of locomotive structures, whether cilia, one cilia, two cilia, numerous, presence of special organelles or cysts as they're formed, and the number of nuclei. Some can have more than one nuclei, some have multiple. We use to identify the different protozoans. Medical specimens taken from human fluids, feces, vaginal discharge, are smeared directly onto a slide and observed with or without special stains. Some are cultivated on artificial media or in laboratory animals for further identification or study. So we can take them, look at them, and a lot of times just by their shape or morphology, we can identify what type of protozoan it is. Okay. Pathogenic disease-causing flagellates. Trypanosomes. Trypanosome is an African sleeping sickness and Chagas disease. T. brucea or T. cruzi. They're long crescent-shaped cells with a single flagellum. Okay. They occur in the blood during infections, and they're transmitted by blood-sucking vectors. Blood-sucking vectors could be ticks, could be mosquitoes. It sucks blood from one species and transfers to another, or one of the same species to another. Okay. From cats, you can have the tick. The infective trypanosome can be infected into humans. Okay. From animals like rats. Okay. You can get ticks that feed on them. It's like an insect vector. So it can travel from the ticks to different species, from other ticks, over the bugs to different species, back to humans. Okay. Infective amoebas. Okay, it's one of your pathogens. entamoeba histolytica is amoebic dysentery. Okay? Very bad digestive disease. Ingested cysts germinate in the small intestine. So you ingest those cysts and then they grow in the small intestine. So they're ingested as benign cysts because they're not active. They're basically hibernating. They come in contact in the small intestine, open up, and become active. Trophozoites migrate to the large intestine and they start to grow there. They can invade your liver, your lungs, and your skin. Gastrointestinal disturbances is a common symptom of it as it grows in the large intestine. Protozoans, okay, there's cysts in food and water. They're not growing. They get ingested into the stomach, into the small intestine, okay, where they start to become mature. We turn trophozoites. They travel to the large intestine, which is where you start infection, causing gastrointestinal problems. Which of the following descriptions is true of both algae and protozoa? They're both photosynthetic. They are both always unicellular. They both contain mitochondria. They're both heterotrophs.
D, they're both heterotrophs.
They both contain mitochondria. They have to contain mitochondria. Remember, things that have mitochondria produce energy to move. Okay? Some algae is photosynthetic, protozoa are not. Okay? Unicellular, some can aggregate, like algae, can aggregate into colonies and into multicellular groups. And then they're both are not heterotrophs. Okay, but they both have to contain mitochondria to provide ATP. Parasitic helminths. These are parasitic worms. Okay, helminths. These are parasitic worms. They're multicellular animals, have organs for reproduction, digestion, movement, and protection. They're parasitize host tissues. They invade your host and they act as parasites. They have mouth parts for intestines or digestion of host tissues, so you can eat your own tissues. Most have well-developed sex organs. They produce eggs and sperm. Fertilized eggs go through a larval period in or out of the host body. Okay. You have flatworms, which are very flat, no definitive body cavity, digestive tract, blind pouch, simple excretory and nervous system. These are tapeworms. Okay. Trematodes or flukes are flat non-segmental worms with sucking mouth parts that grab onto something or can digest tissues. Roundworms, okay, versus flatworms. So tapeworm is a flatworm, just a tube, absorbs nutrients. Okay. And they can be really long, and they do move around the prolactic system. I deployed with the Marines into Ecuador. It was the first humanitarian mission into Ecuador. We took 4 dentists, 12 physicians, no, 8 physicians, four dentists, 4 physicians, and 12 corpsmen. And then we took the entire 7th Marines to get us through the jungles of Ecuador by the Colombian border because of the drug trade. So we went through the jungles for 8 weeks, and we treated people that never saw a dentist, never seen a physician. And we worked on these people, what we could do. Okay. And one of my first patients was this, I'd say 12-year-old kid, and he was choking. He was, coughing up something. So I opened his mouth and I saw something stuck in the back of his throat. So I took cotton pliers, grabbed it and pulled it out. I pulled out a tapeworm it had to be 3 feet long. That came out it was working its way up from his intestines. And he just started choking on it. So these things are.
How does that even happen?
I know. Well, all the foods are infected. When we got back, we had to be dewormed based on all this stuff because you get it when you're over there. You can't drink the water. You can't eat the food. I mean, the first 3 days we were there, the Air Force was supposed to provide water for us, and they didn't show up. So all we could drink there was they had bottled Coca-Cola and beer. That's what was bottled.
That's all I would be drinking the whole time.
Yeah, the Coca-Cola was you couldn't drink the Coca-Cola because it's made from cane sugar over there. So it tasted like pure sugar. It was disgusting to drink. So
okay, then beer it is.
Yeah, you did. I learned una cervezas por favor. Was one more, please. So you learned Spanish over there. I learned abre la boca and cierra la boca, which means open your mouth and close your mouth. But these kids we saw, these people, just black teeth from the Coca-Cola there. They eat sugar cane. Sugar cane is pure sugar. It is crazy, but their teeth were just rotted to the cores. We took out over 3,000 teeth in the 8 weeks we were in the jungles.
I have a question. So what if you wouldn't have extracted that tapeworm? Would he have died? Because it just keeps metastasizing in his body?
I'm sure there were more of them, but it would have just worked its way up until it caused major problems. And we saw a lot of kids with diseases that you can't treat, and they were going to die from it. There's all kinds of disease. Third world countries, it's amazing what you see. Connective tissue disorders, things we can't treat, sarcoplasmas, which are bone cancers. We had portable X-ray machines. You can see their starburst pattern X-rays. You can't treat those. We had a kid that had pneumonia. So we gave the parent the antibiotic. The parent goes, I'm going to sell the antibiotic. I said, well, your kid will die. He needs antibiotic. He goes, I can't have it as a kid. I need more money. I'm going, it's not how we think. It's a different type of mentality over there. It really is.
Wow.
It's amazing what some of these people live through. We saw so many bad teeth. Okay, and physicians treated so many different boils and infections and all kinds of stuff we treated over there. It was an interesting experience. I wouldn't do it again, but it was an interesting experience. I'm glad I went over there, but I was really, really glad to be on American soil where I could drink a big glass of cold water. That's my favorite thing. All right, so your flatworms are your tapeworms, which do grow in the body. Roundworms, okay? These are round versus flat. They have a complete digestive tract. They have a lumen, a protective surface. They have spines and hooks on their mouths so they can anchor. They have an excretory system where they divide nutrients and excrete their waste materials, and they have a nervous system. It's not lungs and respiration and all that, but it's a nervous system that allows it to function and move and respond to its environment. So these worms, okay, helminths, classified according to their shape, their size, their organ development, whether they have hooks or not, whether they have suckers or not, and different specialized structures. How they reproduce, their host, appearance of eggs and larvae. Okay, they're identified by microscope, detections by larvae or eggs, detection of the worm, if you have worms or not. So this is the immature eggs. These are the fertile eggs. These little suckers that adhere to your digestive tract inside your body. And you can see this thing just adheres here and it continues to grow. It absorbs all the nutrients of the food you eat and continues to grow. And if it's motile, it can work its way up. You can start crawling up your stomach, up through your esophagus, and you can actually see it up in your mouth.
Is that a tapeworm there being displayed?
Yeah, this is a tapeworm. Okay. This has an oral sucker right here where it adheres to it. And it looks just like it has a pharynx, an esophagus, an intestine. Okay, has a uterus and a vas deferens, male and female parts for sexual reproduction. Has an ovary, seminal receptacle for sperm, and testes. And then an excretory bladder for urination, get rid of the waste. So it has male and female parts in the same organism. So the genetic material is mixed as they reproduce, so you get varied response or continue evolving. Okay, it's a parasitic worm. Approximately 50 species affect humans. They're worldwide. They occur everywhere. Some are in certain geographic regions with higher incidence in the tropics, like Ecuador. Acquired through ingestion of the larva or eggs in food from the soil or water. Some are carried by insect vectors. They affect billions of humans. Okay. The insects in the tropics, like Ecuador, can be really nasty. They have an ant that flies. It's a black ant, about the size of a big ant, but it lands on you. If you squish it, it releases an acid and burns through your skin, which will go all the way down to the bone. Nasty infection. So you have to flick them off. You can't squish them down. If it walks on you, it will release acid as it walks and leaves a track through your skin. We had 2 people that were medvaced out because of these ants.
Crazy, I knew that.
It is crazy out there what they have. All kinds of parasites, all worms. You got to figure everybody there has worms. You need the food. You can't eat the food. You can't drink the water.
We're really lucky to live where we
We're- Yeah, you don't appreciate what you got until you do. I was real glad I did that. For 8 weeks, spent in the jungles there. I would not want to do it again. But you learn a lot and you appreciate what you have in this country. You really do. Like a pinworm, okay, cross infection between kids, auto inoculation, and kick the neck like cells through the soil or through water. You get the fertile egg, the egg can attach in the mouth, and then you get the infection can spread. And then it has a mouth, it has eggs, has a female part, a male part, and an anus. Okay. So it reproduces inside the body. And it's spread just by oral contamination. Just like the herpes virus is spread through like preschoolers. Parasitic helminths reproduce by, how do they reproduce? Cysts, spores, or eggs and sperm?
eggs.
Eggs and sperm, right? That's how they reproduce. Could be in the same worm. Doesn't have to be two separate ones. Could be the same thing, but genetic material is being mixed.